Hello everyone. Again, I extend a very warm welcome to all of you for this plant physiology and metabolism course. In this lecture, we are going to study the CAM plants and factors affecting the process of photosynthesis. So, you see that we have already studied the light reaction. In light reaction, we have studied the Z pathway, and then C3 plants we have studied in detail. Then C4 plants we have also studied in detail in my previous lectures. So now what are the CAM plants, and then finally we will discuss what are the different factors, both external as well as internal, that affects the process of photosynthesis. So you see that this is the brief overview of the content what we are going to learn today. That is, what is Crassulacean acid metabolism, then which plant come under the category of CAM plants, then what is the basically significance of CAM plants, then how light and carbon dioxide compensation point affect the process of photosynthesis, then finally the difference prevailing among C3, C4 and CAM plants, and then finally the factors affecting the photosynthesis we will discuss in detail, both external as well as internal. Now come to the CAM pathway. You see that some plants adapted to arid environments like cactus, pineapples, which basically employ Crassulacean acid metabolism pathway to reduce photorespiration. Basically this CAM, the name CAM is derived from the family Crassulaceae, where scientists initially identified this photosynthetic pathway. CAM plants, rather than segregating the light dependent reactions and the Kelvin cycle especially, they basically opt to separate these processes temporarily. So you see that what happens in this pathway during the night, what happens, CAM plants open their stomata, permitting carbon dioxide to diffuse into the leaves, and then carbon dioxide is then fixed into oxaloacetate by the enzyme phosphoenol pyruvate carboxylase, employing the same initial step as in C4 plants. Subsequently, what happens, the oxaloacetate is converted into malate or another organic acid. The organic acid is stored inside the vacuoles during the night, and during the day CAM plants keep their stomata closed but are still capable of photosynthesis. So basically this is facilitated by transport of organic acids out of the vacuole, where they are broken down to release carbon dioxide. So this released carbon dioxide then enters the Kelvin cycle for further utilization. By controlling the release of organic acids, CAM plant basically maintain a high concentration of carbon dioxide around the rubisco, ensuring efficient carbon fixation despite closed stomata. So this is the niche CAM plants have over the other plants. Now we will discuss into the detail of the basic mechanism of CAM plants, how this pathway operates. So basically what happens, phosphoenol pyruvate carboxylase is a very pivotal enzyme in CAM pathway, which catalyzes the carboxylation of phosphoenol pyruvate to generate oxalo state within mesophyll cells. So subsequently oxalo state transform into malic acid or other organic acids, which are then stored within vacuoles as a reservoir of carbon. But you see that what happens during the daytime. You see that on the left side of this, you see this is the daytime stomata closed. Sorry, on the right side, during the daytime stomata is closed, and during dark stomata is open. So you see that these during the daytime CAM plants exhibit stomatal closure as a sort of water conserving mechanism. This closure prevents excessive water loss through the transpiration, particularly in arid environment or dry environment. Then what happens within the chloroplast, malic acid stored in the vacuoles undergoes a sort of decarboxylation, leading to the release of carbon dioxide as a byproduct. So you see in the cycle, this carbon dioxide, released carbon dioxide is then further assimilated into the Kelvin-Benzel cycle, where it is utilized in conjunction with other substrate to further synthesize carbohydrates, ultimately supporting the plant's growth and metabolic requirements. The CAM pathway, while being an efficient mitigator of photorespiration, does require ATP as various steps similar to C4 photosynthesis, and this form of photosynthesis is little bit energy costly. However, plants using CAM photosynthesis benefits from enhanced water efficiency. So this is the edge CAM plants generally have. By opening their stomata exclusively at night, you see on the right side, stomata is open. There is when humidity levels are high during night and temperature are little bit cooler or lower, CAM plants minimize basically water loss from their leaves. So this nocturnal opening or opening at the night of stomata reduces the risk of desiccation while allowing for uptake of carbon dioxide. So this is a very special mechanism. You see that in the night stomata are open, whereas in the daytime stomata are closed. As a result, CAM plants basically could thrive in extremely hot and arid environments such as deserts where water availability is very limited. So their ability to conserve water while still conducting the photosynthesis makes them well adapted to survive and flourish in harsh ecological conditions. Therefore, such plants are also known as succulent plants, fat plants, because the storage of water in their leaves and stem give them a swollen or fleshy appearance. That's why fat plants. The best known succulent plants you might have observed in your garden is cacti. Now, how these CAM plants are distributed among the families? So, they are very much prevalent, and about 24 families are the angiosperm flowering plants such as Crassulaceae, Partulaceae, Cactaceae, Euphorbiaceae, Bromilaceae, Azoceae, Agaveaceae and Orchidaceae that contains CAM metabolism. While most of these families, basically excluding Crassulaceae and Cactaceae, doesn't solely rely on CAM, as they have members using both CAM and C3 photosynthesis. So in this respect, these two families are little bit different from most other families which I mentioned. So some plants might even have plants utilizing all the three modes of photosynthesis, that is C3, C4 and CAM. Commercially important plants that exhibit CAM includes pineapple, agave, cacti and orchids. In addition to flowering plant, you see CAM plants also occur in certain ferns such as iceates, some lithophytic and epiphytic ferns, and in Velvetchia mirabilis, a gymnosperm you might have heard. CAM pathway is also observed in certain aquatic plant species. So this is little bit bizarre, and you see that certain aquatic plants also possess CAM pathway. Certain CAM plants like cacti are considered obligatory CAM users, while other succulents show a facultative CAM strategy. So one is obligatory CAM users, another one is facultative CAM users. A very notable example of a well investigated facultative CAM plant is the plant of Mesembryanthemum cristolinum. So this is annual plant and belonging to family Azoceae. It is very interesting that when there is a plenty of water available, this plant behaves similarly to a conventional C3 plant. However, when the plant faces water scarcity or high salt level, it switches over to CAM pathway. Now, what is the significance of CAM pathway? So, we will discuss what are the adaptations that made CAM plants very effective to carry out photosynthesis. So, as I mentioned earlier that CAM plants, they are well suited to dry places like deserts, have a unique ability to grow and attain biomass even when there is a little rain or lot of evaporation. So basically these plants, they are able to do and survive because they open their stomata at night when it's cooler and less water is lost through the process of transpiration. So this makes or you can say this allows them to take in carbon dioxide from the air. During day, you see that when the stomata are closed, they use basically carbon dioxide from the night to carry out photosynthesis. The transpiration ratio, that is the ratio of water transpired to the ratio of weight of water transpired to the weight of dry matter produced is very important for CAM plant and they have subsequently lower than either with C3 or C4 plants. So in this respect, CAM plants differ from C3 and C4 plant, and CAM plants basically use water much more efficiently than other type of plants. For you see, for every gram of carbon dioxide they gain, they lose only about 50 to 100 grams of water for gaining 1 gram of carbon dioxide. Whereas C4 plants lose around 250 to 300 grams and C3 plants lose about 400 to 500 grams for the same amount of carbon dioxide, that is 1 gram. This makes CAM plants better suited to dry environments like deserts where water is scarce. Now in terms of carbon assimilation rate, CAM plants doesn't assimilate carbon as quickly as C3 or C plants generally do. Even though they are efficient with water, they can only take in about half as much carbon each day as C3 plants and about 1/3 as much as C4 plant. Interestingly, CAM plants are not only found in CAM plant, they are not found only in deserts. Some aquatic plants also use CAM, just I mentioned earlier. And scientists are of opinion that you see that CAM plants basically help them to take carbon dioxide when there is not much carbon dioxide available in the water. This happens when there is a lot of resistance to gas diffusion, making it generally very hard for carbon dioxide to move around freely. So the closure of stomata during the day in CAM plants, which helps them to save water in dry environments, might not be only reason why CAM plants evolve. This is because more surprisingly, some plants that live in water, that is in aquatic condition, also show CAM behavior. Some scientists even believe that CAM plant might have evolved to help plants in water environment acquire inorganic carbon in the form of bicarbonate ions when there is not much carbon dioxide available. This happens when there is a lot of resistance to the movement of gases, making it very hard for carbon dioxide to move freely in the water. Then we will discuss evolutionary process, how CAM plants have evolutionary significance. So you see that CAM plants, they, they are thought to be much evolved. We will discuss then light compensation point. So in dark condition, photosynthetic carbon dioxide fixation does not occur, and plants release carbon dioxide due to aerobic respiration. In the presence of light, photosynthetic carbon fixation begins. So when light intensity increases, photosynthetic carbon dioxide fixation also increases. This is a very interesting point you must note here. At a certain light intensity, it matches with the uh, carbon dioxide released through the process of respiration. So at this point, this is very crucial. So this is the point which is known as light compensation point, where basically carbon dioxide uptake by photosynthesis equals to carbon dioxide released by the process of respiration. So no net carbon dioxide fixation occurs at this compensation point. So the light compensation point varies with the plant species and the various developmental conditions. Generally, the low compensation point is indicative of photosynthetic efficiency, as the plant is then using maximum amount of available carbon dioxide. Shade plants typically have lower light compensation point than plants grown in sunlight due to their lower respiration rates. So this is the reason. Next we will discuss what is carbon dioxide compensation point. Similar to light compensation point, the carbon dioxide compensation point is reached when the amount of carbon dioxide fixed by photosynthesis equals to the amount of carbon dioxide released by respiration. So this is very interesting. At this point, there is no net release of carbon dioxide from the plant. This concept is like the light compensation point just I discussed earlier, but there is the focus in the carbon dioxide compensation point on the balance between the photosynthesis and respiration based on carbon dioxide concentration rather than light intensity. So for C3 plant, the carbon dioxide compensation point is typically between 30 to 70 ppm at 25 degrees C. C4 plants have much lower carbon dioxide compensation point, often less than 10 ppm, due to their carbon dioxide concentrating mechanism. So in this respect, carbon dioxide C4 plants have an edge over the C3 plants. The actual concentration basically at which the actual carbon dioxide concentration at which compensation point varies with environmental factors, especially temperature and among different plant species. For example, you see that C4 plants like maize, sorghum and sugarcane have extremely low carbon dioxide compensation point, that is about 2 to 5 ppm. What does it mean? It means they can almost completely use up surrounding carbon dioxide. Whereas on the other hand, you see that C3 plants like tomato, wheat and soybean uh, have higher carbon dioxide compensation point, about 50 ppm or more, which can increase with higher temperature due to increased rate of photorespiration, due to increased rates of photorespiration. Now, this slide shows the comparative account of differences among C3, C4 and CAM plants. You see that there are number of points on which C3, C4 and CAM plant differ from each other, and I have mentioned these points while discussing C3 plant, C4 plant and CAM plants, but going all these differences in one go will further strengthen your concept in what respect these three pathways differ from each other. Now, after discussing all the aspects of photosynthesis, we should know that what are the factors, external factors and internal factors that affect the process of photosynthesis, or you can say the rate of photosynthesis. So, you see that the rate of photosynthesis can be influenced by various factors, both external and internal. So, it is very important to consider the principle of limiting factor at this juncture. And now, what are the principle of limiting factor? According to this principle, that the rate of any physiological process such as photosynthesis is limited by the factor that is the shortest supply relative to the demand. So, in other words, it could be mentioned the rate of a process is generally determined not by the total amount of resources available, but by the most limited resource. So, it is very interesting. So, if any factor is below its optimal level, it will limit the overall rate of process regardless of the abundance of other factors. So, basically this principle helps to explain how multiple factors such as light, temperature, carbon dioxide concentration interact to determine the rate of photosynthesis. Now, you see on the slide that there are different factors like light, light intensity, carbon dioxide concentration, temperature, water and oxygen. So, we will discuss one by one how these factors affect the rate of photosynthesis. First external factor is light. So, sunlight is the you see that the primary source of light for photosynthesis in green plants. Sun is the only source of light for green plants, but moonlight can also stimulate photosynthesis in marine algae. Even artificial lights like electric light can also trigger photosynthesis as long as it is within the visible part of the spectrum. So, rate of photosynthesis is basically also affected by quality of light in different ways, like photosynthesis, as you know, that primarily occurs in the visible spectrum, and chlorophyll absorbs maximum in the red part of the spectrum, leading to maximum photosynthesis in red light. The next highest rate of photosynthesis occurs in the blue light, while rate of photosynthesis is minimum in green light. In addition to this, photosynthetic rate is higher in white light such as daylight compared to monochromatic light of a specific color. Now, light intensity. So light intensity basically differentially affects the rate of photosynthesis. Typically photosynthesis occurs more efficiently in intense light compared to diffuse light. This very contrasting figure. However, certain plants known as zeophytes, these plants thrive in shady environments where they can achieve optimal photosynthesis within less intense light. Conversely, heliophytes are the plants that require uh, intense sunlight to maximize their photosynthetic process and are commonly found in sunny areas. It's important to here mention here that excessive intense light can hinder the process of photosynthesis, a phenomena known as solarization. This solarization basically occurs when cellular components including the photosynthetic apparatus undergo photooxidation, which leads to death of the plant. Additionally, extreme intense light can cause other limiting factors that come into play, thereby reducing the rate of photosynthesis. Coming to the duration of light exposure, even a brief flash of light is adequate for photosynthesis to occur. However, photosynthetic rates tends to be higher in intermittent light compared to continuous light, we have studied in our previous lecture. This is because in continuous light, what happens, assimilatory power accumulates and is not utilized in the dark reaction as rapidly as it is produced in the light reaction. Now, how carbon dioxide affects the process of photosynthesis? Carbon dioxide is generally present in the environment atmosphere by about 0.03%, or you can say 330 ppm by volume. So an increase in carbon dioxide concentration up to around 1% can enhance the rate of photosynthesis. However, excessively high concentration of carbon dioxide can be toxic and lead to a decrease in the rate of photosynthesis. Then temperature also plays a very crucial role in the process of photosynthesis, affecting both the rate of efficiency of photosynthesis process in different plants. Basically, different plants show varying temperature requirement for photosynthesis. For example, many plants stop photosynthesis around freezing point. Certain conifers can even continue the photosynthetic process even at temperature as low as minus 35 degrees C. So, if we look at the other side, uh, and during extreme temperatures more than 40 to 5 degrees C typically inhibits photosynthesis in most of the plants. However, some xerophytes, like as I mentioned earlier, Apantia and algae could thrive in hot springs, and photosynthesis even can uh, be operative at extreme temperature of 55 and 75 degrees C. Usually there is a notable enhancement in the rate of photosynthesis as the temperature rise from 10 degrees C to approximately 40 degrees C. So, this temperature sensitivity is basically denoted by temperature coefficient for photosynthesis, which is usually around 2. Then water. Water serves as a very crucial raw material and very important factor for photosynthesis. But you see that water scarcity rarely serves as a limiting factor for photosynthesis. Inadequate water supply can indeed decrease the rate of photosynthesis in different plants. And during the process of photosynthesis, water molecules are split in a process known as photolysis, providing electrons to replace those lost in the light reactions of photosynthesis. This process is basically essential for generation of oxygen and synthesis of ATP and NADH, which are very vital for the subsequent synthesis of glucose in the Kelvin cycle. So, in this way water is very important and crucial. Now, last factor is oxygen. The presence of oxygen generally can inhibit the rate of photosynthesis, a phenomenon known as Warburg effect was This was first observed by German biochemist Otto Warburg. What happens in Warburg effect, that oxygen basically you see that competes with carbon dioxide for the active site of the enzyme Rubisco during the Kelvin cycle, reducing the efficiency of carbon dioxide fixation and thereby limits the process of photosynthesis. This competitive inhibition by oxygen decreases the photosynthetic activity, particularly in high oxygen concentration when availability of carbon dioxide is limited. So, it is very important to mention here, note here that Rubisco exhibits both carboxylation and oxygenation activities. However, under high oxygen levels, Rubisco becomes more reactive with oxygen due to the changes in its kinetic properties. So, notably you see that temperature affects gas solubility and carbon dioxide becoming less soluble at higher temperatures, whereas O2 is less affected. Now coming to the internal factors, which also plays very crucial role. Chlorophyll content, you see that is very important for plants to do photosynthesis. In non-green plant, part of the variegated leaves are in etiolated plants. Generally, photosynthesis does not take place. However, you see that some scientists disagree whether there is a direct link between how much chlorophyll and how fast photosynthesis happens. But if we talk about it logically, it makes a sense that if there is a more chlorophyll, photosynthesis should happen faster, as long as every else the plant need is also favorable. Now, second factor that affects the process of photosynthesis is protoplasmic factors. So, you see that proper hydration of the protoplasm is really very important for photosynthesis. But even if you take out the part of the plant cell that do photosynthesis, for example chloroplast, and give them the right conditions, they can still do photosynthesis on their own. Now, accumulation of the end products of photosynthesis also affect the process. Generally, when photosynthesizing cells start to store a sort of carbohydrate, it actually slows down the rate of photosynthesis. But if these carbohydrates or the end products of photosynthesis are quickly translocated to other parts of the plant, it can actually help to speed up the rate of photosynthesis. Now, anatomy of the leaf is also very important. The rate at which photosynthesis happens in any leaf or other photosynthesizing part of the plant is generally affected by the anatomy of the leaf or the plant part. The things like how thick the outer cuticle and the epidermis are, how the stomata are arranged, where and how much of the green chlorophyll containing tissue is, and how the vascular tissue is set up, all play a role in the rate of photosynthesis. So, this must be clear. The internal structure of the leaves or other plant parts where photosynthesis occurs mainly affects the rate of photosynthesis by influencing how much light can get into the chlorophyll containing cells, maintenance of the trajectory of those cells, and moving to the sugars made during the photosynthesis out of those cells through the vascular tissues. Then, of course, microstructure of the chloroplast is very important. The rate of photosynthesis can be affected by the structure of chloroplast. For example, C4 plants contain two types of chloroplast, while C3 plants have only one type of chloroplast. Therefore, microstructure of the chloroplast, including the presence of an organization of the different chloroplast types, can influence the efficiency of photosynthesis. So, we have studied in detail the CAM pathway, that what are the specific features that CAM plants have, then the other pathways C4 and C3 plants. We have also studied the factors affecting the process of photosynthesis in detail. So you see that overall photosynthesis holds immense significance for life on Earth. That it produces oxygen, provides energy, and serve as the foundation of food chain. So thank you very much for today's lecture. Now, hello everyone, I am Qazi Faruddin. Extend a very warm welcome to all of you for this course. So in today's lecture, we are going to dive into the very fascinating word of mineral nutrition in plants. Now, what are the aspects we are likely to cover in today's lecture? What are the mineral nutrients? Then, what are the different criteria that must be adhered to consider an element to be essential? Then, what are the different criteria adopted for classification of essential mineral nutrients? Then, different criteria, different classification will be discussed in detail. So, what is mineral nutrition in plants? So, once you see that after being absorbed from the roots, the mineral elements are translocated to different parts of the plants and they serve in numerous biological functions. So, the study how plants obtain and use mineral nutrients is known as mineral nutrition. So, basically plants derive essential nutrients from the three main sources: air, water and soil. Non-mineral elements such as carbon, oxygen and hydrogen are obtained from the air. These elements are basically very crucial for photosynthesis and other metabolic processes within the plant, just we have gone through in our previous lectures. The mineral elements are basically derived from the soil in inorganic form. These minerals are absorbed by roots, and due to their large surface area and their ability to absorb inorganic ions at low concentration from soil solution, generally its effectivity increases, uh, effectivity of mineral acquisition increases by the plants due to large surface area. And once these essential mineral elements are absorbed by the roots, they are further translocated to various parts of the plant where they serve as a diverse biological functions. These functions include enzyme cofactors, structural components you might be aware, and in addition to this, regulators of various metabolic processes. So, the acquisition and distribution of mineral nutrients are very crucial process that sustain the health and vitality of the plants. So, in addition to roots, other organisms such as mycorrhizal fungi and nitrogen fixing bacteria often play very crucial roles in the acquisition of mineral nutrients by plants. These symbiotic relationships basically enhance nutrient uptake efficiency and contribute to overall health and productivity of the plant. So, to study how plants obtain and utilize mineral nutrients is uh, known as mineral nutrition. So, basically this field of research is very crucial for improving modern agriculture practices and environmental protections. In addition to this, this study helps in understanding the complex ecological interactions of plant within natural ecosystem. So, the application of mineral nutrients primarily through fertilization is very crucial for achieving high agricultural yields. Indeed, the yields of most of the crop plants show a linear increase with the amount of fertilizer they absorb. Basically, due to this, the importance of proper nutrient management in maximizing crop productivity is very crucial. So, at present, to meet the growing demand for food, you see the annual world consumption of mineral nutrient used in fertilizer, especially nitrogen, potassium, and phosphorus, has it, increased, and you see that from 30 million metric tons in 1990, in 1960, it increased to 143 metric ton in 1990. And in recent years, this annual consumption of mineral nutrients has further scaled, reaching to 180 million metric tons. So, this trend underscores the significance of nutrient supplementation in modern agriculture to further sustain the global food production. Now you see that half of the energy used in agriculture is generally devoted to production, distribution, and application of nitrogen fertilizers. In addition to this, the production of phosphorus fertilizer is likely to reach its peak during this century. However, despite the substantial investment in fertilizers, crop plants utilize less than half of the nutrients applied to the soil around them. So, utilization efficiency is at present at how much? Only 50%. So, what would be the fate of the rest of the mineral which is not taken up by the plants? So, the excess minerals from the fertilizer can have very detrimental effects, as they may leach into the surface water or groundwater, or adhere to soil particles, or contribute to air pollution and climate change. However, generally plants offer a beacon of hope and serve as a traditional means of recycling and removing these deleterious materials from the environment, owing to the complex relationship between the plant, science, and the atmosphere. So, the study of basically mineral nutrition is a multidisciplinary field involving various experts of plant sciences like atmospheric chemist, soil scientist, uh, microbiologist, ecologist, and plant physiologist. All these experts contribute to further understanding the intricate processes that are involved in nutrient uptake and utilization by plants. So, we can say that the collaborative efforts are basically required for developing sustainable agricultural practices and mitigating the environmental impacts associated with mineral nutrient management. Now, you see that what are the basic criteria that must be adhered for an element to be classified as essential mineral elements. So, only few elements have been determined to be essential for the plant growth. The basic concept of essentiality of mineral nutrient was proposed by Arnan and Stout in the year 1939. So, basically they identified 16 elements as essential for plant nutrition, and so for an element to be considered an essential nutrient, it must meet a specific criteria. So, what are these criteria? Number one, deficiency of an essential element must prevent the completion of the life cycle of the plants and result in deficiency symptom. So, plant will not be able to complete its life cycle in the absence of a particular element. Next criteria is that essential elements cannot be substituted by another element with similar properties, and the essential element plays direct roles in plant metabolism. In the absence of essential elements, plants are unable to uh, produce viable seeds. So, you see that uh, another criteria is essential elements should also be component of essential plant metabolite, like magnesium is a constituent of chlorophyll molecules. So, these criterion set by Annan and Stout is a very specific criterion set almost 80 years back that must be adhered to classify an element to be essential. Now, how these essential mineral elements are classified? So, the first criteria of classification of essential mineral elements is on the basis of relative concentration in the plant tissues. So, uh, on the basis of relative concentration in plant tissues, all the essential mineral nutrients could be classified into two categories. One is macronutrients, another one is micronutrients. You might be aware. So, what are now question arise, what are these macronutrients? Macronutrients, they are also known as macroelements, are the mineral elements required by the plants in large quantities, and they are typically found in plant tissue in concentration ranging from 0.2 to 4% on dry mass basis. So, you see that their concentration is high, and then these macronutrients, they are further categorized into two types: primary macronutrients, which consist of nitrogen, phosphorus and potassium, and secondary macronutrients, which include calcium, sulfur, magnesium and silicon. Basically, potassium, calcium and magnesium are generally grouped together as they are predominantly present as cation. Likewise, nitrogen, potassium and sulfur, they are grouped together because they are primarily present as anions like nitrate, sulfate and phosphate. Now, moving onward, the next category is micronutrients. You see, they are also referred to as microelements, and these are the mineral nutrients. They are found in the plant tissue in smaller quantities, typically measured in ppm, ranging from 5 to 200 ppm, or less than 0.2% of the dry mass. These micronutrients are minerals include boron, chlorine, manganese, iron, zinc, copper, molybdenum, sodium and nickel. And these microelements, they exist in various forms including inorganic ions, oxyanions, or undissociated molecules of boron, as well as in organic compounds complex known as chelates. So, you see this table shows the classification of mineral elements based on their concentration, and basically the tissue levels of essential elements required by most plants are mentioned in ppm. In this table, you just go the required mineral nutrient in plant, or you can say their concentration present in the plant tissue. Now, you see that what happens in certain specific cases, the differences in the tissue concentration between macro and micronutrients may not be very significant, as we have seen our just previous table. For example, the mesophyll in leaves of in the mesophyll cells that contains almost as much as iron or manganese, that contains the leaves of the mesophyll tissues of the leaves of the plant contain uh, iron or manganese just almost equal to sulfur or magnesium. So you see that these elements, these microelements are frequently found in higher quantities what than the micronutrients. So some of the researchers generally argued that a classification based on the concentration, that is macronutrients and micronutrients, is difficult to justify physiologically. So scientists namely Konrad Mengel and Ernest Kirkby in the year 2001 classified essential elements on the basis of their physiological and biochemical roles. So in this table you see that the classification of plant nutrients could be categorized into 4 different major groups based on their physiological roles and biochemical functions. So you see that in group 1 there are only 2 nutrients, essential nutrients, nitrogen and sulfur. So these nutrients are basically, they are the integral components of carbon compounds like nitrogen, which plays a very crucial role as a constituent in various organic molecules. And nitrogen also serves as a very fundamental components of amino acids, amides, protein, nucleic acids, nucleotides and coenzymes. It is also present as hexamines and other essential biochemical compounds. And you see in the table another element present in the group first is sulfur, which is also a very crucial element in carbon containing compounds, and it serves as a constituent of cysteine and methionine. It's a very important point to remember here, which are essential amino acids. Moreover, you see that sulfur is also involved in the structure of lipoic acid, coenzyme A, thymine pyrophosphate, glutathione, biotin, 5-adenyl sulfate and 3-phosphoadenosine. So you see that both nitrogen and sulfur contribute significantly to the composition and functionality of numerous carbon-based compounds essential for carrying out various biological functions. Now we see in the second group there are 3 essential mineral elements, namely phosphorus, silicon and boron, and these are second group of elements. They are essential elements and play a very crucial role in the energy storage or structural integrity within the biological systems. Like you see, phosphorus is very important molecule involved in energy storage and structural support in the plant cell. It is present, you see that in the sugars, phosphate, nucleic acids, nucleotides, coenzymes, phospholipids and phytic acid. In addition to this, phosphorus plays a key role in reactions that involve ATP, that is the primary energy currency in the cells. Another element present in the second group is silicon, and this silicon is basically deposited as amorphous silica within cell walls. This element contributes significantly to the mechanical properties of cell walls, enhancing rigidity and elasticity. That is also very important. Hence the presence of, you see, the silicon is very crucial for maintaining the structural integrity of the cells and tissues. The third element present in the second group is group 2 is boron, and basically boron complexes with various compounds such as mannitol, mannan and polymanuric acid, which are constituents of cell wall. And in addition to this, boron is involved in processes related to cell elongation and nucleic acid metabolism. It is present, the presence of boron is very much essential for the proper cell growth and development, as well as maintaining the structural integrity of the cell walls. Now group third nutrients generally contains potassium, calcium, magnesium, chloride, zinc and sodium, and these group 3 element nutrients, they are involved uh, and they are generally in ionic form within the biological system. Like you see that potassium, which the first element in the group third, serves as a cofactor for more than 40 enzymes and principal cation responsible for establishing cell target pressure and maintaining cell electroneutrality. And calcium also plays a very crucial role in cell wall structure, which serves as a constituent of middle lamella. It is also required as a cofactor for enzyme involving ATP and phospholipid hydrolysis and acts as a secondary messenger in metabolic regulation. And you see that magnesium is essential for activity of numerous enzymes involved in phosphate transfer, and it is also constituent of chlorophyll molecule. We have discussed in while discussing the photosynthesis. Then chlorine. Chlorine is basically necessary for photosynthetic reactions involved in oxygen evolution, contributing to the production of oxygen during photosynthesis. Now next element present in the group third is zinc, which serves as a constituent of various enzymes including alcohol dehydrogenase, glutamic dehydrogenase and carbonic anhydrase, playing very essential role in metabolic processes. Sodium. Sodium is also involved in cellular processes like other essential mineral elements and plays a key role in the regeneration of phosphoenol pyruvate in C4 and CAM plants, and it can also substitute potassium in some functions. So in this respect, sodium has a very important role. Now group 4, these essential elements, they are basically play crucial roles in redox reaction within the biological system. Like iron serves as a constituent of cytochromes and non-heme iron proteins involved in vital processes such as photosynthesis, nitrogen fixation and respiration. Manganese is basically required for the activity of various enzymes including dehydrogenases, decarboxylases, kinases, oxidases and peroxidases. And in addition to this, manganese also plays a role in photosynthetic oxygen evolution. Now next very important uh, element present in the group 4 is copper, which is a component of numerous enzymes involved in reactions including ascorbic acid oxidase, tyronase, tyrone- tyrosinase, monoamine oxidase, uricase, cytochrome oxidase, phenolase, lacase and plastocyanin. So the presence of copper is very crucial for electron transfer process and maintaining cellular redox balance. So other element that comes under group 4 is nickel, which is constituent of urease and hydrogenase involved in nitrogen fixing bacteria and playing essential role in metabolism of nitrogen. Next last element that comes in group 4 is molybdenum, which is a constituent of various enzymes such as nitrogenase, nitrate reductase and xanthine dehydrogenase. These enzymes are basically involved in crucial redox reactions including nitrogen fixation and nitrogen metabolism. Now, another way to classify essential mineral elements is on the basis of their mobility. So, some elements they are mobile and some are immobile. So, you see in this slide that on the left side there are number of elements that comes under the category of mobile elements, namely nitrogen, potassium, magnesium, phosphorus, chlorine, sodium, zinc and molybdenum, whereas immobile elements include calcium, sulfur, iron, boron and copper. So as far as mobile elements are concerned, they are transported by the plant's vascular system, allowing for redistribution from older to younger tissues as required. So basically, this mobility enables plant to prioritize the allocation of nutrients, ensuring the essential functions are maintained even under varying environmental conditions. So you see that what happens, the deficiency symptoms appear first in older leaves when nutrient reserves are depleted to support the growth and development of new tissues. Now, in contrast to mobile elements, certain elements exhibit limited mobility within plants. Like you see in the slide, this list include boron, iron, calcium, and copper. So once they are absorbed by the roots, these immobile elements, they tend to remain localized within the tissue where they are initially taken up. Consequently, deficiency symptoms for immobile elements are typically observed first in younger leaves where nutrient uptake occurs, as these nutrients are not easily translocated to other parts of the plants. So you see, in this aspect, immobile elements are very important because their deficiency symptoms will be reflected in younger part. So basically, the precise mechanism of nutrient mobilization are not very well defined. Uh, there are different plant growth hormones which could be involved for translocation of these mineral elements like cytokinin. So this table shows basically the relative distribution of various elements in earth crust, and most of the elements just mentioned in different color, like in the periodic table you see a specific color is given for essential mineral elements that could be identified, and then in addition to essential mineral elements, they are beneficial elements. So beneficial for some plants but not required by other plants. So on the basis of mobility, initial symptoms, deficiency symptoms could be visually observed. If uh, visual symptoms are initially observed in the older parts, this deficiency symptoms could be due to the mobile elements. If initial deficiency symptoms of any essential mineral element is observed in the younger part, it is sure that it could be due to the deficiency symptom of immobile elements. So initially both the deficiency symptoms, on the basis of mobility of the deficiency symptoms, uh, it's very easy to uh, observe that this plant is deficient due to the deficiency of mobile elements or immobile elements. So this is all about my today's lecture. We have gone through the complete classification of essential mineral elements, first on the basis of concentration, then on the basis of physiological roles and biochemical functions, and third on the basis of mobility in the plants. So after finishing this lecture, you must be able to identify whether this deficiency symptom is due to the mobile elements or immobile elements. And in my coming lectures, I will discuss in detail about their function, uptake, function, and deficiency symptom of specific essential mineral elements. Thank you very much.